thank you for the introduction. So my talk is about the common pitfalls in clinical metabolomics, but most of the samples will be actually for lipidomics, and I will give you a guidance for the sample collection as well as how to target the data analysis problems. So first, let me give you a short overview about clinical investigations. I mean, everyone who was involved in them probably knows that they can be quite complex and require expertise in many different areas. And today I want to focus on two specific problems, because I would say that the LC-MS analytics, probably most of you already have a solution here, maybe a biocrates kit or your own assay. But today, so I want to focus on the pre-analytics and the dataset integration, because they are mostly overlooked and also always at the border between two areas of expertise, in this case, like the clinicians and the analytics department, and here for the analytics department and the data scientists mostly. So first, maybe let me explain what is pre-analytics referring to. So everything basically that happens before the actual analytics, which can have an impact on the compound concentrations, is considered pre-analytics. And of course, the nutrition and exercise status can be have a high impact on the metabolite or lipid profile, but I think they are quite hard to harmonize. But which can be harmonized quite well and has also a great impact on the lipid and metabolite concentrations is how you process the samples, especially the processing time and temperature, especially those for plot samples. So for those who do not know so much about pre-analytics yet, I can highly recommend this review by Rainer Lehmann. It gives a great overview about all the pre-analytical factors which are involved in plot sampling and also some different matrices. And he also gives some quite good recommendations on how you could address these factors. But to make it really easy, you can also look at the recommendations by the Lipidomics Standards Initiative. So they give recommendations on several different topics, and they also provide some best practices how you should prepare your lipid samples. And you should always collect and prepare the samples on ice and immediately flash freeze them and then store them at less than -80 degrees. So this protocol basically works for all matrices, and that was what it was intended to do. And from my personal experience, I can actually confirm that this is a good protocol for lipid analysis. But in a clinical setting, is it really feasible? Especially the direct sample processing and flash freezing? I think we have some issues here. So, but then why is it important in the first place? I was really interested that someone gave a talk, and he was commercially interested in S1P, but he said that they would measure several different stuff, but they would not measure the concentrations of S1P because the quantification was considered not reliable. So if maybe several of you are already analyzing lipids, so we might know it. I think it's not a quantification problem, it's a sampling problem. Because if you take a plasma or whole blood samples and store them at room temperature before centrifugation, after just 20 minutes of incubation time, you get about a 90% increase in S1P concentrations. And of course, this can greatly impact your results. The S1P is presumably released from platelets, but there are several publications stating that S1P is probably a good biomarker for whatever disease, but they were not referring to how the samples were processed, and some of them even used serum samples, which always requires incubation time at room temperature. Of course, none of these researchers intended any ill will, they just didn't know. But for the future, of course, we should try to avoid this bias. But how? Because we are actually interested in more than just one compound, not just S1P. There are so many other lipids and metabolites we want to look at, and as you might see, it gets a little bit more complicated if you try to take a look at the whole picture. So those are the results for more than 400 lipids and polar metabolites. But you can also see that there's good news in here, because many of the results are lightly colored, meaning that those compounds are actually considered to be stable. You can further confirm that the Lipidomics Standards Initiative recommendations of cooling your samples actually does improve your stability. And you can further consider that the removal of the blood cells also improves your stability, because then they can't release any lipids or modify them in some way. you can further see that there are some class-specific relations, so that you see those clusters here. Because like in many other areas, also in paralytics, several compound lipids act in a lipid class-dependent manner. For example, the endocannabinoids, the sphingoside base 1 phosphates, which I mentioned before, but also lysophosphatidic acids and also other lysophospholipid. But to make it a little bit more complicated, there are also some compounds which kind of act out of order. For example, the 12-HT is not stable, whereas other HT appear to be stable even at room temperature. And of course, there are also some other polar metabolites which fall out of this schema. So what do we do now? If we can't get those best practice results and those really fast sample processing? Because I think it's then quite difficult to assess which compounds are stable and which are not. So to maybe make this a little bit easier, we then tried to define different pre-analytical protocols which could be used in different clinical settings. For this, we then tried to first think of which protocol would make sense in most clinical settings, also in a study which encompasses different study sites. And for this, we thought that 60 minutes each was a suitable time. So we have some collaborators which also managed to do the complete sample processing until freezing in just 20 minutes, including 10 minutes of centrifugation. But this is just one study set and have to be really, really on time for this. So 60 minutes, I think, should be feasible. And then we tried to loosen those restrictions as much as possible without losing too many compounds. And we actually could extend the time to 4 hours each without losing too many compounds due to instability issues. But of course, this is under cooled conditions. If you now take the same protocols and store the samples at room temperature, we actually might lose quite a lot of compounds. And also, you should consider that there might be some lipids, like the endocannabinoids, which are not stable at all under these conditions. So to assess the stability, we used a really simple fold change approach based on like a 20% cutoff, which is also like mostly considered a suitable RSD or CB for the QC samples in lipidomics. So for a whole blood and a plasma each, it should be less than 20%, and in combination, it should be less than 30%. So for prospective sampling, you might be able to fit one of these protocols. But what do you do if you already have taken your samples and the protocol kind of doesn't match, if it's like a combination of different settings or maybe other compounds which you did not investigate in the study? And to make this a little bit easier, we then tried to aggregate our data as well as some data from the literature which was available into a database which can then be accessed using our Shiny app. So we called it Alistair, the application for lipid stability evaluation in research. And the main idea of this application is that you could answer tailored pre-analytical questions at different time points in your analysis. Of course, at best, you would already consider your pre-analytics before actually taking the samples when talking to the clinicians. Here you could try to interact with them and showing them if you prepare the samples like this, or then you lose those compounds which could be important for the study. So you might be able to motivate them more to generate high quality samples for you. But if you have already taken your samples, maybe from a biobank, you can still enter your specific pre-analytic conditions and then assess which compounds would be stable, so which compounds would make sense to analyze. So in case you have different assays and you want to save some money here. If you have already analyzed all of the compounds, maybe you just use the extended MXP quant kit and have just analyzed all of them. You could also use it then to filter the data so that you only use high quality compounds or compounds which can be detected with high quality from a pre-analytical perspective into your data evaluation to already filter out some compounds. And of course, in the end, you can look for specific lipids or polar metabolites to make sure that your potential biomarker is not just maybe a pre-analytical artifact. So maybe to make this a little bit more relatable for you, I then have here an example. So using the data filtering mode, I try to upload then the compound list of the extended MXP quant kit. And here then you can, and then I entered some pre-analytical conditions, which I seem to just feasible. And here you can see then the status, the label for each of these compounds. And the first thing you will actually realize that most of them are gray, which means they did not find a match in the database. How can that be? The extended MSP Quant kit actually has more than 1,000 compounds, but so does our database. So how can it be that there are so little of an overlap? And here, this is actually then the perfect transition to the next problem of the dataset integration, because I think the overlap is actually quite high. But of course, the nomenclature of the compounds could be different, and that then can cause that the database can't match these compounds. So then I tried again, renaming some of the lipids. So the Alistair already encompasses a Gosling parsing algorithm. I will come to that a little bit later. But I then also had to calculate the sum of the fatty acid side chains, because if you might know, the MXP Quant kit uses MS for the detection, and it can already resolve, for example, one of the fatty acids in the D and triglycerides. So this can be useful information, but in Alistair, as there's not so much available on these compounds, it only supports the total sum of the fatty acids. So then by recalculating this, I actually then got quite a better match. It was still not perfect, but probably because we now have some duplicates in these data sets. But now you can actually kind of get a better pre-analytical insight, because if you now compare two different conditions, for example, like the preferred conditions with less than 60 minutes for the processing each on ice, you see that there are actually some compounds which are not stable, some which might be questionable, but in general it looks okay. But now if you take, for example, 2 hours of processing time at room temperature each, you see that the number of unstable compounds significantly increases. And that can be then really useful, because if you don't consider pre-analytics, maybe your samples, or probably they will not be taken in a really short time. So, but now let's get back to what this integration of the datasets means. So in our lab specifically, this problem is a little bit larger because we use a combination of so many different assays. We encompass the non-targeted analysis using high-resolution mass spectrometry for the screening of the abundant lipids and polymetabolites, but we further complement this with LC-MS analytics with absolute quantification for the lowly concentrated lipid mediators. But of course, using all these assays, we get also some kind of a compound overlap. For example, ceramides are important lipid mediators, but they are also quite abundant lipids, so they're also covered in the lipid screening. And of course, now the question is, how do we tackle this compound overlap, also in assays which use different quality criteria and different selectivity? So first with the selectivity. Probably all of you know that in mass spectrometry, and especially in lipidomics, it's really important that the nomenclature you use to label your compounds should reflect your level of identification. And now if you look at the 5 levels of identification proposed by Szymanski et al., for lipids you already see some difficulties, because you might have an MS-MS match confirming that this should be the right structure. But several lipids have more like lipid class-specific MS-MS spectra, so it's not actually compound-specific. And maybe you even have a reference standard to confirm the retention time as well as a spectra, but are there isomers? Because if there are isomers, you basically should buy all of these isomers, have reference standards for all of them, and then actually prove that all of them are separated using zero chromatographic resolution. Otherwise, you cannot confirm that it's exactly this compound. And for data evaluation, especially if you're going to path analysis, this can be really difficult, because a biologist usually wants to have a specific compound, but for lipidomics, mostly that's not possible. So one way, one approach to solve this was the lipidomics shorthand nomenclature. I can highly recommend to use this to label your lipids, because it reflects your identification level quite well. And there's also the Gosling algorithm, which can be used for automatic name parsing and maybe translating some names from, let's say, older publications or something, and to assist you in this. Still, it probably requires some further knowledge to correctly apply this nomenclature, as there are some difficulties in this. For example, if you take some bile acids like the cholic acid or taurocholic acid, for the cholic acid, you actually have to assume that it could also be an oxidized sterol, whereas for taurocholic acid, it's sure it must be a bile acid. And if you're just looking at this from the outside, how should you know how many isomers exist, whether there is a sterol isomer which you could also detect? So in the details, it might be difficult to apply this correctly. Also, for example, for the sterol esters, in mammals, you could actually assume that most of the sterol esters are cholesterol esters because 95% of them contain cholesterol. But then, do you label it how you actually can detect it as a sterol ester, or do you just assume that it's cholesterol esters? And of course, this does not work at all for polymetabolites. Trust me, I tried. It doesn't work. And as mentioned before, pathway analysis tools, they don't like the shorthand nomenclature because it's not specific enough. There's one exception, and that is Linux. I will come to this a little bit later. Of course, the metabolomics community also has a solution for this, so sort of. The RefMet nomenclature, it technically also considers different annotation levels or identification levels, but sadly, in the automatic name parsing, which it provides, it is not reflected at all. Sometimes it actually even adds some structural information which was not present before, as in this example with the aconitic acid, where it's then also adding the cis, but you can't know if it's a trans-aconitic acid. And last but not least, a problem I think that will be really important for you if you're using the biocrates kits or in general, is the overlap with, in general, data sets with different selectivity. And in our lab, for example, we might analyze an oxidized arachidonic acid in the lipid screening, but we have 7 different heat resolved in a lipid mediator assay. And probably kind of the problem in the Alistair example I showed you was that in the extended MXP quant kit, the XL kit, you measure up to 10 isomers of TG56-7. And of course, all of these can provide different information and might act differently. So this is then a huge problem. How do you handle this data? Because Alistair is unfortunately not alone in the tools which only require a specific level of identification. The other tool I mentioned before was Linux, which is a really nice tool for pathway analysis for lipids, because it does not look at specific enzymes, but rather in lipid-specific or class-specific reactions, like the deletion or addition of fatty acids or head groups, or the modification of the said fatty acids or head groups. But here, then, if you have then an assay which does not have either the total sum of the fatties or a total resolution of all fatty acids, the software unfortunately will not accept it. So here you actually then have to lose a little bit of information by calculating the sum of your lipids. And this might work quite well if you already have analyzed the sum, or I mean, for example, if you take the PCs, you got some quite interesting results, and then you can look at the subclusters and get a really better insight into which compounds to look at. But what you then should consider that if you have a bias in between or some limitation in your in the method, like in this case that you had to lose some selectivity in order to use this application, you should consider that it might not give you the best results you can get. Because in another study I unfortunately cannot show you today, we actually measured with the MXP kit, the normal kit, the 500 kit, and we tried to use Linux. But interestingly, the triglycerides which were interesting from a different statistical approach were not covered with Linux, or so they were not detected. There was nothing interesting about them, and it was most likely that the software couldn't handle that there was several isomers then now aggregated kind of together. So unfortunately, I don't have a good solution for you. I can only recommend you to consider always the limitations of your tools and try to always try to combine different approach, because every data set is unique, and what might work really great for one data set might turn a blind eye on a different data set. So use those tools, but also like maybe take a more specific look at your data, and maybe just something as simple as a radial plot like this can also give you some informations, because like if you have one fatty acids in the TGs resolved, this actually already gives you some information. And here it might also be interesting to not just to look at those compounds here which are significantly increased, but maybe also those which are not. Like in this case, it's always like the O16-1, which is not significantly increased, and that could also be some interesting information. Sadly, the problems don't stop here, because you also have to consider the accuracy of your quantification. And in the lipidomics area, there are several different quantification strategies, and I think all of them have their valid points and their valid applications. But now, what do you do when you have one data set or one study with different assays, with different quality criteria, different quantification approaches? So in this case, we have an example of our one of our COVID studies where we used the MX3-Quant500 kit, but we also complemented it with LC-MS analysis of some lipid mediators and our own HRMS screening method. So at first, this might sound redundant, but actually those assays complemented each other. But of course, we had several duplications in compounds, and how do you address them? I mean, the first point is, of course, always to try to match the nomenclature so that you actually catch it if there is a duplicate in compound. But then we try to look at the quantification strategy. And in this case, we just try to order them according to what we presume would be their accuracy. So the best accuracy we assume with an external calibration or 6-point calibration using our LC-MS assays. And then we considered the FIA MS of the MX3-Quant kit as better than our reversed phase HRMS screening, because here the internal standards are correlating to our compounds, so it is a more stable relative quantification, and also, of course, biocrates has done some validation studies on this. And here then we just excluded all those duplicate compounds to get just one data set, because otherwise, what would you do if you have the compounds in duplicate? Do they correlate? Do they not correlate? I think this might be just a really easy approach. But why does it make sense to look at the quantification in this way? I mean, for those who are more interested to take a closer look into the quantification of lipids in general, I can highly recommend taking a look at this review of the ceramide ring trial. So they investigated the NIST plasma concentrations, or tried to post reference concentrations for NIST plasma of 4 different ceramide compounds using an external calibration as well as a matching internal standard. And it's a really interesting read, because you can already see that even with this, like, let's say, high quality quantification, still many issues occurred, and you have to actually take a closer look. But in general, it does work. And they compared also the results, how they would change if they use a different internal standard than the directly labeled standard. So I tried to recreate this for a little bit more compounds, the ceramides we have in our assay, which each of them has their own labeled internal standard. But then I tried to compare the results using a different internal standard, just like the next matching one kind of, and also try to calculate the concentration if I would just use a 1-point calibration, either with the matching or with the alternative internal standard. And as you can see, the absolute quantification results compared to like what would be the best quantification, they are all over the place. I mean, this looks horrible, kind of. So for absolute quantification, not good. But what they recommended actually in the ring trial was to use a reference sample, for example, the NIST plasma sample, to then normalize your results to improve their robustness. And this actually works quite well. So in this case, I then just used our own control samples. This could also be, for example, the QC samples provided by MX3-Quant kits. And here you then actually see that the results or the relative quantification is much better. So what can you take from this? I think the relative quantification can be really useful, and you can deliver really interesting and exciting results using these. But please avoid illusions about what the absolute quantification of your assay means. I mean, of course, you can try to validate it so that you know how much or how big the error is. Maybe there is not an error. But of course, for an outsider just reading a publication, it might be difficult to assess this. So please try to be honest with this, with yourself and with others. And of course, how do you address this if you have now multiple data sets? I mean, in the biocrates kit, they already do a little bit of the work for you because they label the compound quantification quality, sort of. So they label it whether it's quantitative with some restrictions or whether it's relative quantitative. And what I now propose to you is that then you try to reduce your important features, maybe by doing a correlation analysis or whatever. Don't just take all of the compounds as equal, but actually try to look at how good is the quantification accuracy. Because if you want to then assess a biomarker in the end and want to do a validation study, I think you have much better chances if you are in this group rather than this group. So, of course, don't omit this group, but also try to consider using the more quantitative results first. And of course, if you have additional assays, you might want to add some to these labels. For example, we try to differentiate between compounds which have their own label standard, because as you have seen before, this actually does change a lot for the absolute quantification. And maybe you even have a reduced position for the relative quantification, but if there's just at this point no other way to measure this compound, it could still be useful. But of course, you can extend this list to whatever you like. And maybe if you have already an accredited clinical assay, let's say total TG content, and it's completely correlating with all of your 300 TGs, maybe you just want to stick to the already validated assay and consider this for your future data analysis. So with this, I then want to conclude my talk. So just as a short summary for pre-analytics, for the sample handling, especially processing time and temperature, and really important. And you should always try to keep the samples under cooled conditions and process them as quickly as possible. If that is not possible, you should consider what is impact on your results and discuss it accordingly. For the dataset integration, nibbled nomenclature is really important, but also the quantification is really important. And maybe you want to consider both of them to always kind of select the highest quality of compounds. Because in the end, I think lipidomics in clinical research projects can actually deliver really interesting insights. There's still a chance that we will find those really interesting biomarkers which can help in diagnostic approaches of underdiagnosed diseases, as well as provide some further insights, for example, into the pathophysiological process and maybe some new drug targets. So with this, I want to conclude my talk and thank my working group and of course you for your attention.